In this lecture, we'll talk about stem cells. So we'll learn what are stem cells, what different types of stem cells, and we'll learn about their various applications. Stem cells actually, stem cells these are the cells that has the ability to continuously divide and differentiate into various other kinds of cells and tissues. They have unlimited potential to proliferate and then further differentiate also. So this is a brief history of stem cells. It was first identified in 1981. In embryonic stem cells in mice were identified. 1989, Mario Martin-Evans and Oliver they developed the knockout mouse technology. In 1997, the Dolly the sheep was created by using animal cloning. 1998, isolation of human embryonic stem cells were identified in 1998. 2001, President Bush restricted federal funding for embryonic stem cells research, particularly in case of human embryonic stem cells. 2004, Harvard researchers grew stem cells from embryo using private funding. So when the funding was restricted by U.S. government, so they started private funding. 2006, cells reprogrammed. So this was a major milestone. Actually, these are induced pluripotent stem cells. So stem cells were created. 2007, Nobel Prize Evans shared the Nobel Prize with Mario and Oliver Smith for works on embryonic stem cells. 2010, person with spinal injury became the first to receive a medical treatment derived from human embryonic stem cells. 2012, human embryonic stem cells show medical promise to treat blindness. 2012, Nobel Prize Yamanaka wins Nobel Prize for creating induced pluripotent stem cells. So we'll learn about what are these IPS cells or induced pluripotent cells and how they can be very useful in treating so many diseases in humans. 2014, therapeutic cloning with adult cells. Skin cells from a woman with diabetes were used and demonstrated that the resulting stem cells can be turned into insulin-producing beta cells. So their therapeutic potential of these stem cells is endless actually. So we'll learn in this lecture. 2014, human trials, therapy based on induced pluripotent cells to treat a form of age-related blindness. So these are some of the major milestones in the research on stem cells. So what are the important characteristics to identify stem cells? So they are unspecialized cells. They are not. So they are highly unspecialized cells, and they are capable of dividing, proliferating, and renewing themselves for a long period of time. Proliferation and renewal. So these are another important characteristics of stem cells, and have the potential to give rise to specialized cell types. So okay, so they can differentiate also, further differentiate also whenever there is a requirement in the body. So different types of stem cells, totipotent. Each cells can develop into a new individual cells from early. So these are the totipotent cells are from early embryos, 1 to 3 days old embryo. Pluripotent cells can form any cell types over to around 200 types. Some cells of blastocyst 5 to 14 days old embryo. Multipotent cell differentiated but can form a number of other tissues like fetal tissues, cord blood, and adult stem cells. So these are different types of stem cells. So in case of the fertilized eggs, when it develop into further undergo cell division, it formed blastocyst. So you can get these cells from this after the they are the totipotent stem cells, and when they form blastocyst containing they contain the pluripotent cells here in this stage. So they are the pluripotent cells, and then these blastocysts containing pluripotent stem cells. Then when you isolate them, so they can form different types of cell, hematopoietic cells, tissue, mesenchymal cells, neural cells, or different types of cells they can form. So they become unipotent. So from totipotent, this is embryonic from the fertilized egg. So at this stage they are totipotent, and then they become pluripotent, and then multipotent, and from multipotent they become unipotent. They become highly specialized cells. So there are different types of stem cells again. Embryonic stem cell, 5 to 6 days old embryo. Embryonic germ cell derived from the part of human embryos or fetus that will ultimately produce eggs or sperm. They are embryonic germ cells. Adult stem cell are undifferentiated cells found among specialized or differentiated cells on a tissue or organ after the birth. So these adult stem cells are actually they are undifferentiated cells. They are not differentiated cells. They are undifferentiated cells found among the specialized or differentiated cells in a tissue or organ after the birth. These are that's why we call them adult stem cells. So whenever there is a requirement, these adult stem cells can then get differentiated to form specialized cells. But these cells appear to have a more restricted ability to produce different cell types and to self-renew as compared to embryonic stem cells. So pluripotent stem cells actually they have more potential to become any type of cells. Like from the blastocyst, if you take this pluripotent stem cell, you culture these cells in in vitro conditions. Then from these stem cells you can get different types of differentiated cell like pancreatic cell, hematopoietic, cardiomyocytes, neuron, hepatocytes. Different types of cells can be obtained from these pluripotent stem cells. Multipotent stem cells, actually multipotent stem cells limited in what the cells can do. Like suppose if it is from brain, if you get this stem cells from brain, so they can just develop into neurons or glia cells. If you take from heart, these stem cells, they will develop into cardiac muscle cells. So similarly, so they have limited potential for differentiation. These multipotent stem cells. So this is how embryonic stem cells can be obtained from the after the fertilized eggs start dividing, undergo cell division and all that one. So during in vitro fertilization, these embryonic cells can be obtained. So these are the just to show the stages of embryogenesis. So this is one fertilizer undergo first division. So the cleavage, 8 cell stage, blastocyst, blastocyst inner mass, cell mass, and you can get the from this inner mass cells you can get the pluripotent stem cells. So this is again same thing. Fertilizer egg become totipotent cells. You get blastocyst here. From these blastocyst inner cell mass contain pluripotent stem cells. So this cell isolated from the ICM. Then you can culture them in under in vitro condition in lab. And so you can culture these pluripotent stem cells and then you can use it for further studies. Adult stem cells and these are adult stem cells are actually undifferentiated cells found among specialized or differentiated cells in tissues or organ after the birth. So they are present in skin, fat cell, bone marrow, brain, many other organ and tissues. So these stem cells are present in all, but their number is very low. Like in case of bone marrow, finest bone where blood cells form used to replace damaged or destroyed bone marrow with healthy bone marrow stem cells. Suppose there is some damage in bone marrow, some cells are damaged, so bone marrow stem cell can get differentiated and replace the damaged cells. So these bone marrow stem cells they can treat patient diagnosed with leukemia, anemia, and lymphomas. So this just slides to show how the blood cells formation occur. Hematoblasts, proerythroblasts can get from the hemocytoblasts. Okay, you can get pre-erythroblasts, then polychromatic erythroblasts, and then you get erythrocytes. Similarly, other from these particular stem cells can get differentiated into leukocytes and other different types of lymphocyte monocytes. So these can be from the same stem cells which are present in the bone marrow. Umbilical cord stem cells, also known as Wharton jelly, actually stem cells are infants. So these are the adult stem cells of infant origin after the birth. Okay, in stem cells of infant origin. So less invasive than bone marrow. Okay. So it's easy to obtain actually from this and greater compatibility and less expensive also. So the three important functions of umbilical cord: plasticity, potential to change into other cell types like nerve cells; homing, to travel to the sites of tissue damage, so they can travel to site of the tissue damage and then repair the damaged tissue; and grafting, to unite with other tissues, so they can easily get merged with the other cells. So there is another types of stem cells. So these are called cancer stem cells. So we'll learn a little bit about the cancer stem cell. What are these stem cells actually? In most of the cases, stem cells actually they are useful for the our human body for us. But so there are one type of stem cell. These are called cancer stem cells, which can create a problem in our body. So normal stem cells is the real cells within the organs with the ability to self-renew and give rise to all types of cell within the organ to drive organogenesis. So they are the useful normal stem cells. So they can proliferate and then can get differentiated whenever there is a requirement in the system. Then besides normal stem cells, there is cancer stem cells also. So these are the rare cells within the tumor. So they are not normally not present in normal person, but they are present in the within the tumor. So rare cell within the tumor with the ability to self-renew and give rise to phenotypically diverse tumor cell population to drive tumorigenesis. So their main role is to drive tumorigenesis. They're present in the tumor cells, and these stem cells then give give rise to tumor cells only, and then help in tumorigenesis. So this is just to show again comparison between normal stem cells and the cancer stem cells. So this is just the stem cell, normal stem cells. It can undergo proliferation, undergo cell division and proliferation, and multiply, and then it can get differentiated into different types of cell like neuron, cardiomyocytes, skin cell, okay, different types of cell, hepatocytes, and then we have cancer. So so these are the cancer stem cells. Cancer stem cell will not form these any of these neuronal cells, cardiomyocytes or blood cells and all that one. It won't form. So this pathway will be blocked in cancer stem cells. It will form only tumor cells. It will help in growth of the tumor only. So these are called cancer stem cells. So this is the main difference between a normal stem cells and cancer stem cells. For the first time, the cancer stem cells were observed in the leukemia, blood cancer, but then it was found in other types of cancer also. So CSC's cancer stem cells have recently been identified in several solid tumors also, include cancer of the brain like brain cancer, breast, colon, ovary cancer, pancreatic cancer, prostate, melanoma, multiple myeloma, and non-melanoma skin cancer. So all these different types of cancer now cancer stem cells have been identified and detected and identified also. Okay. So, but first they were identified in a leukemic patient, leukemic patient blood. So there are several theories for the origin of cancer cells, how these cancer stem cells get originated in the body actually. So they are derived basically from the normal stem cells only. Okay, so this is a stem cell here, normal stem cell. Okay, these stem cells can undergo, there can be some genetic or epigenetic changes. Genetic or epigenetic changes can occur, some mutations can occur and all that one. And then it start getting changed. This particular normal stem cells get mutated, and then slowly when it accumulates sufficient amount of mutations, it can get transformed into a cancer stem cell. From normal cancer stem cells can become a cancer stem cell after undergoing several genetic changes, undergoing some mutations or epigenetic changes, DNA methylation and all that one. It can get converted to cancer stem cells. Similarly, there is a possibility that the differentiated cells, okay, due to some genetic or again through some mutations or epigenetic changes, it can got de-differentiated. Okay, de-differentiated, and then it get stemness renewed, means it become more like a stem cell, normal stem cell, and then it get converted into cancer stem cell. So these are some of the theory for the hypothesis for origin of cancer stem cells. So basically they are derived from a normal stem cells also, and through mutations and other epigenetic changes they become cancer stem cells. So like suppose this is a primary tumor here. Okay, and here you see a red color type cell, hair cells. So this is highly tumorigenic. So basically this is a cancer stem cells. So this particular cancer stem cell which is highly tumorigenic. It has unlimited self-renewal activity at the helm of tumor hierarchy, can be resistant to conventional therapies and to other treatment. And the other cells in the tumor actually, they are poorly tumorigenic and they have limited self-renewal activity. So here this is the other cells have limited capability for proliferation and all that one. But this is highly tumorigenic. So this is the one actually which create a lot of problem because they are resistant to many chemotherapy drugs and all that one. But these can be killed. These can be taken care of because they have limited self-renewal activity. But they are these are the one highly tumorigenic. These are the cancer stem cells. So they are highly tumorigenic, and then they can form new tumor cells. So if you isolate these one and then again grow, so they can again form tumors in the body. So they are the one which can create spread, which can help in metastasis or the spread of the cancer in other parts of the body. So how does this cancer stem cell create problem actually? Like suppose here is a tumor here, and in this tumor, because the cancer stem cell, like just like normal stem cells, cancer stem cells, are population of cancer stem cells is also very low in the tissues. So their number is very low. So once that few so once the treatment is carried out, because most of the chemotherapy drugs and other treatment, they kill the normal tumor cells and they don't target the stem cells basically. So more, because most of the cancer drugs have been developed to target the normal cells, normal cancer cells, not the stem cells. So when the patient is given these drugs, so they will kill the tumor cells like other tumor cells, but not the cancer stem cells. So cancer stem cells will survive the chemotherapy treatment. So in this, so once the drugs is given, tumor will shrink because most of the cells will normal cancer cells will die, but the cancer stem cells, they evade the treatment, so they will escape this chemotherapeutic drugs, and then again they will slowly with time so they will again proliferate and give rise to the same tumor. So this is how the cancer relapses even after the treatment. So much of treatment, the cancer relapses in many patients, cancer patients. So now how to get the complete cure actually in this situation? We need to develop some drugs which can target the cancer stem cells also. So now suppose there is a tumor and we have cancer stem cells here. So now you have the drugs for both which can target these normal cancer cells, which can target the tumor, and also which can target the stem cells, that kill tumor cells, stem cells also. And then tumor loses its ability to generate new cells, and slowly it will, both the drugs will then target tumor as well as the cancer stem cells, and that tumor will shrink, and the tumor and the cancer will be completely cured. So there won't be any relapse of the cancer. So this is how the now now the most of the therapies are being targeted uh, to take care of cancer stem cells also, so that there is no chance of cancer relapse. So this actually, the most therapy failed to consider the difference in drug sensitivity of cancer stem cells compared to their non-tumorigenic progeny. Most therapies target rapidly proliferating non-tumorigenic cells and spare the relatively quiescent cancer stem cells. So most of the therapies actually, drugs have been developed to target only the tumor cells, not the cancer stem cells. So cancer stem cells escapes in many situations. after treatment, but then again they divide and then form tumor again. So now to completely treat cancer, we need to develop some drugs which can target the cancer stem cells also, so we can get a complete recovery from the patient can get complete recovery from the cancer. So this was an example of cancer stem cell, but normal stem cells have now several application in medical field. So these can be used for tissue repair, any tissue repair like nerve, heart, muscles, organ, skin. They can be used for cancer also, cancer treatment, autoimmune diseases like diabetes, particularly therefore diabetes, arthritis, MS. So these can be used. Stem cells can be used for treatment of several diseases. Like tissue repair, like they can be used to regenerate spinal cord. As you know, as I told, these stem cells can form can proliferate and then differentiate into different types of cells like heart cardiomyocytes, neuronal cells and all that one. So they can be those, after differentiation, those cells can be used then to regenerate spinal cord, heart tissue, or any other major tissues in the body. They can be used to replace skin, damaged skin, burnt skin also. Heart disease like adult bone marrow stem cells injected into the hearts are believed to improve heart cardiac function in victims of heart failures or heart attack. So these stem cells can play a major role in controlling heart diseases. Like suppose, so this is an example how the stem cell can help in improving the condition of heart. So in this case, actually what they have done here, mouse adult stem cells are injected into the muscles of the damaged left ventricular wall of the mouse heart. Stem cells then help regenerate damaged heart muscle. When these stem cells are injected, the green one here you can see here, when they are injected, these stem cells help regenerate the damaged heart muscle cells. Okay, so they can repair. So these stem cells then can form new blood vessels, formation of new blood formation in the damaged heart muscles, and proliferation of ancillary vasculature, and then the heart muscle, that damaged heart can be then repaired using these stems. So this can be used the stem cells can be used in treating of cancer, like particularly case of leukemia. Leukemia can be so the leukemia patient treated with stem cell emerge free of disease. Particularly in the case of leukemia, stem cells can be used to cure leukemia. Injection of stem cell have also reduces pancreatic cancer in some patients. Arthritis, rheumatoid arthritis, also stem cells may be helpful in repair of the eroded cartilage. The cartilage damage, so these adult stem cells can be used. Type 1 diabetes, it can be also cured. Pancreas, in type 1 diabetes, pancreas cells do not produce insulin. So now embryonic stem cells might be trained to become pancreatic islet cells needed to secrete insulin. So embryonic stem cells can be taken and then they can be differentiated into pancreatic cells and then injected. So they will secrete the insulin, and then type 1 diabetes can be easily tackled. So this is just an example of adult stem cells in the adult brain. Here you see some blue in this picture. Actually, here you see some blue dots here. So they are basically the adult stem cells in the brain. So when you culture these blue cells, adult stem cells, then they can get differentiated into different types of cell like astrocytes, neuronal cells, oligodendrocytes here in the picture actually. So basically this is just to show that stem cells are present in adult brain also, and they basically play a role in repairing the damaged neuronal cells and other types of cells, astrocytes and glia cells. So these blue dots, when they when they were isolated, these stem cells were isolated and then cultured in vitro, they started getting differentiated into different types of cells. Like blue cells, here is the stem cells here, and some of them got then differentiated into neuronal cells, astrocytes or oligodendrocytes, glia cells. So this is just to show that the adult stem cells are there in our adult tissues, and then they are used in the body for replacing the damaged cells. So now their application of these stem cells is tremendous. It can be used, as I mentioned in earlier slides, they have tremendous application in treating so many diseases. But the problem is to get these stem cells, because it's not ethical to get these cells from the fetus. Okay, so now this one technique was developed by Japanese scientist Yamanaka. Actually, he he reprogrammed the cells. Basically, differentiated cells he reprogrammed them and converted them into stem cells. So these were called IPS cells, or induced pluripotent stem cells. So once you have these IPS, then there is no ethical issue. So we don't have to get the stem cell from the fetus and other earlier embryonic stages of the human. So these cells can be easily created in the lab. So how do we create actually? So we'll go through this few slides, and you can learn how to make these IPS cells. So here one can take the normal cells, okay, isolate cells from the patient skin or fibroblasts grow in a dish, because from the patient actually it's very easy to collect the skin cells. It's known in not much painful or not in that much difficult or invasive. So the skin cells can be easily isolated from the patient, and then you can culture them. And then these cells, after these cells were treated with reprogramming factors. So we'll learn what are these reprogramming factors actually. So these cells were treated with reprogramming factors, and after few weeks, the these skin cell, as you know, they are differentiated cells, adult differentiated cells, and after reprogramming, they become pluripotent stem cells. Okay, so they got, after a few weeks, after the treatment with these factors, this differentiated skin cells became pluripotent stem cells. So now we got the pluripotent stem cells. Now once you have these stem cells, then you can stimulate these stem cells to differentiate any variety of cell type. Suppose if you need muscle cells, you can make muscle cells. You make heart cells, cardiomyocytes, you can make that one. Or you need blood cells also, you can convert them into blood cells. So we can just change the culture condition and differentiate these pluripotent stem cells, any type of cells of your choice or your desire, whatever is the requirement, so they can be differentiated. So now this is easy way now of getting the stem cells. So these are induced pluripotent stem cells. You can form any type of cell, neuronal cells, okay, dopaminergic neuronal cells, motor neuron, cardiac muscles, hematopoietic cells, blood cells, any type of skin cells, any type of cells can be then formed from these induced pluripotent stem cells. Okay, so now these are stem cells which were developed in the lab. So that's why they are called induced pluripotent stem cells. So how do we? So what are those reprogramming factors actually? So they were actually the different types of genes, regulatory genes actually. So these Yamanaka group, Takashi and Yamanaka group in 2006 did this pioneering work actually of they developed this in IPS cells, induced pluripotent stem cells. So they identified 24 different genes. 24 genes which play very important role, say key role actually in maintenance of embryonic stem cells in embryonic stem cells in the body. Okay, so these are the different genes here. Okay, so these 24, so they tried these 24 genes, different combination of genes, and they started putting these into in these with the differential cells. So they treated 24 they treated this they introduced 24 different combination of these 24 genes into these skin cells and converted it into pluripotent stem cells. So these are the list of the 24 different genes. Some of these important genes for actually this expression of OCT34, SOX2, KLF4, CMYC, and factor essentially required. So they are the important genes which is required for maintenance of stem cells. So they divided into few groups, three groups actually, which are responsible for pluripotency. Group 1 actually ES cell specific transcription factor. So they are the genes actually which are expressed in, these are the transcription factors which play a very important role in maintaining embryonic cells. So ES cell specific transcription factor, they are essential for pluripotency in embryonic cells and early embryo. So they are expressed during embryonic stage only in embryonic stem cells, and they are useful in that one in maintaining pluripotency. So OCT34, SOX2, NANONC, so they are the important transcription genes. In group 2 is proto-oncogenes, important for proto-oncogenes. Basically, they are involved in proliferation, important for proliferation of embryonic stem cells, but not in early embryos. TLC1, STAT3, CMYC, ERAS, KLF4. Okay, so these two group are very important. And then group 3 actually expressed in embryonic stem cell but less defined function, ECT, ESG. So these are the three. They divided these 24 genes into three groups, and group 1 was basically to maintain pluripotency, and group 2 was mainly for proliferation. So these OCT3, OCT4 involved in the maintenance of self-renewal of pluripotent cells. Repression in immunity cell leads to formation of trophobactoderm. Overexpression leads to formation of various lineages including primitive endoderm. So basically this is important gene here. SOX2 actually essential for embryonic development. Downregulation by silencing leads to the differentiation of cells in embryonic stem cells. So this is very critical during embryonic development. So other genes, important genes are KLF4. So another one is in nano actually for it is also involved in promoting pluripotency. Lin28 is an mRNA binding protein expressed in embryonic stem cell and embryonic carcinoma cell associated with differentiation and proliferation. So these are some of the important genes which are critical. So these genes basically which encode for transcription factor can be introduced into any adult cells, and then those adult cells can be then converted into induced pluripotent stem cells. So in the production of IPS, so one has to transfect these genes into adult cells. So this is IPS so transfection of certain stem cell associated genes into non-proliferating non-pluripotent cells such as the fibroblasts, skin cells, and others. Transfection is typically achieved through viral vector like retrovirus, and this after 3 to 4 small number of transfected cells begin to become morphologically and biochemically similar to pluripotent cells and are typically isolated through morphological selection, doubling time, or through a reporter gene or antibiotic selection. So this is how the IPSs, induced pluripotent stem cells, are created. So this is again the same thing. Isolate and culture donor cells like skin cells, transfect stem cell associated gene into basically the all different combination of transcription factors which are basically stem cell associated gene into the cells by viral vectors. Here You introduce these in harvest and culture the cells according to AS cells culture. So they get transformed. Some of the cells will get once they are transfected with the transcription factor or the embryonic stem cells related genes. They become they develop stem like embryonic stem like characteristics. So they are then isolated, and small subset of these transfected cells will then become iPS cells and generate like ES like colonies, like embryonic stem cell like colonies here. So this is how the iPS induced pluripotent stem cells are created. iPS cells, they are regarded as a holy grail for stem cell research, studying disease models in vitro, drug screening. They can be used for drug screening, particularly to see that toxicity of drugs in during embryonic stages and all that one. Toxicology testing of new drugs, generating patient-specific and disease-specific pluripotent stem cells. So this is the most important one. Here they can be used for generating patient-specific or and disease-specific pluripotent stem cell for treatment of various diseases. Allow unprecedented access to all stages of human biology, studying development and function of human tissues, regenerative medicines. So they have now a lot of applications basically. So this is one application how iPS cells can be used to cure anemia, sickle cell anemia. So like suppose this is a mouse with sickle cell anemia. So we collect the skin cell from this mouse. Okay, so they're cultured in vitro, and then you put the embryonic cell specific transcription factor like OCT4, SOX2, KLF4, c-MYC, okay, uh, using viral vectors, and then these skin cells are reprogrammed into iPS cells. Okay, now once we have the iPS cells, okay, then one can do the gene editing, or by gene editing one can do the we can correct the mutation, whatever is the mutation causing sickle cell anemia, it can be corrected, and then these genetically corrected iPS cells can be taken and then proliferated, and then you can differentiate them into blood stem cells, and then once these you have the blood stem cell, then you can inject into the mouse and replace the old blood cells with the new one. So they will proliferate, and then one can cure the sickle cell anemia in patient. So this is the how the stem cell iPS cells, basically iPS cells can be used to cure various diseases. So this is one example actually. iPS can be used for treatment of Parkinson disease also. Cell replacement therapy seems particularly suitable for Parkinson disease, a common neurodegenerative disease caused by loss of midbrain dopamine neurons. So the dopamine neurons are basically damaged or so here, so they can be replaced. Transplanted fetal midbrain cells has been shown to restore dopamine function in animal models and in human patients. Even the mouse kidneys have been created using iPS cells. A team of scientists has successfully used induced pluripotent cells to create kidneys inside a mouse whose parents were genetically injured to so that their offspring would not be born with the organ. So this also can be achieved. Actually, one can develop tissues also. So mouse kidney. So now here the somatic cells, basically the in iPS cells they can be used to generate beta cells also. Pancreatic axon cells can be converted into beta cells closely related to the iron beta cells, can be used to cure diabetes also. So there will be some technical challenges like cell line may have mutations, so one has to be sure that there is no mutations when the cells are selected. Delivery to target areas, so wherever there is a requirement, target area should be delivered there, and should be rejection prevention or rejection. But if it is taken from the patient, so that issue won't occur actually if the cells are taken from the patient. then will be easy. So we'll give another example how actually these stem cells can be used for treating some known curable diseases. So there is a disease called critical limb ischemia. Critical limb ischemia is the term for severe blockage of arteries of the legs, leading to reduced blood flow. So there is a blockage in the arteries which supply blood to the legs, so which leads to reduced blood flow. And gray form of peripheral arterial disease, actually CLI, is a result of atherosclerosis or the arterial hardening and narrowing due to arterial plaques or fatty deposit in the arteries. Here there is a blockage actually in the arteries, so it leads to critical limb ischemia. Critical limb ischemia or CLI is a severe obstruction of the arteries which seriously decreases blood flow to the extremities, particularly hand, feet and legs. Severe obstruction of blood flow results in ischemic rest pain, ulcer or gangrene, and a significant risk for limb loss. So this, once the blood supply is reduced here in the legs, it leads to formation of gangrene. So this is the critical limb ischemia case. So because of the poor blood supply, cells start getting damaged here, and this and then it leads to gangrene and complete, and then there is the risk of limb loss also. So some symptoms are actually this one: pain in the legs and feet when the person is at rest and not moving. So this is one symptom, actually pain in the legs and feet when the person is at the rest. Pain is felt that time. Source of infection of the legs and feet that won't heal. Okay, healing is wound healing is reduced. Faint or absent pulse in the legs or foot below the blockage. So there is also so these are some of the symptoms of critical limb ischemia. So now these stem cells can be used for treatment of critical limb ischemia, for which there is actually no drug at present. So now there is the only hope for this treatment of the critical limb ischemia is stem cells now. So the collection, so here now the bone marrow collection, the step 1 is bone marrow collection. The collection of bone marrow is done from the hip bone of the patient by using a thin needle under the local anesthesia here. The stem cells from the collected bone marrow are then evaluated to measure their strength and vitality. The next step is implantation of stem cells in the target area of the patient, either by angiography or okay. So then these stem cells can be then implanted into the target area, particularly the hand and feet, wherever there is a tissue damage. So this is how the stem cells can be used for treatment of critical limb ischemia. So here, besides bone marrow, collection of bone marrow, one can take from the skin cells also, skin cell of the patient, and then they can convert them into induced pluripotent stem cells also. Okay, so both way can be done. So this is one method, collection bone marrow cells and then injected into the damaged tissues, where then it can replace the and can develop the new blood vessels, and the blood supply can be restored in that damaged tissue. So this is how the critical limb ischemia can be cured. So basically, once the treatment is done, improvement in one can see the repair of the damaged tissue, and one can observe the pain at the rest, whether it is during resting there is no pain at all, and so that can be checked if the therapy is working. So stem cell application are tremendous now. You can see tissue repair, nerve, heart, muscles, organs, skin, all these can be repaired. Cancer study shows leukemic patient treated with stem cell emerge free of disease. So particularly for leukemic patient, this is a boon actually. Cancer stem, stem cells are boon. They can be used for treating leukemic patient. Autoimmune diseases like diabetes, rheumatoid arthritis, neurodegenerative disease, Parkinson disease also can be cured using stem cells. So therapeutic application and endless for stem cells. And now we have iPS cells, so induced pluripotent stem cells. So it can be used quite easily. I mean, there's stem cells are now easily available because of iPS cells now. So why the stem cell research is so important? Stem cells can replace disease or damaged cells. Stem cell allow us to study development and genetics. Okay. Then besides the stem cell can be used to test various drugs and chemicals to see their toxicity or how safe these drugs are during for the embryos. So there is some controversy if these embryonic stem cells derived from the blastocyst or from the fetus. Okay, so there's but now because of IPSS now we don't have to worry about these ethical issues.